Good day, it's Dr. Will, CBMD PhD case, Thursday, June 30th, 2016. It is 1:06 p.m. Eastern Standard Time. Hematology definitions. Guanine purine found in all living organisms is a component base of DNA and RNA. Phosphate, any salt or ester of phosphoric acid. Guanosine triphosphate, a nucleotide, the 5-transpose triphosphate guanosine. It is an activated precursor in the synthesis of ribonucleic acid. It is acid involved in energy metabolism being produced from GDP by substrate-level phosphorylation in the tricarboxylic acid Krebs cycle, and serving as a source of free energy to drive protein synthesis. The ratio of the JTP and ATP is maintained by the reversible transfer of phosphate catalyzed by GDP kinase. Guanosine, a purine nucleotide, guanine linked by the N9 nitrogen to the CI carbon of ribose, is a component of ribonucleic acid, and nucleotides play important roles in metabolism, symbol G. Trimeric, exhibiting the characteristics of a trimer. Trimer, a compound formed by combinations of 3 identical simpler molecules, 2 a capsomere having 3 structural units. Inhibit, hinder, restrain, or prevent an action or progress, person from acting freely. Enzyme, protein producing by living cells and functioning as a catalyst in a specific biochemical. Juxtaposition, apposition. Heterotrimeric, a trimer having at least one subunit that differs from the other. Cytosolic, pertaining or contained in the cytosol. Cytosol, the liquid medium of the cytoplasm, i.e. cytoplasm minus organelles and non-membranous insoluble components. Hydrolyzed, subject to hydrolysis. Hydrolysis, lysis, dissolution, the splitting of a compound into fragments by the addition of water, the hydroxyl group being incorporated in one fragment and the hydrogen atom in the other. Intrinsic, inherent, essential, belonging naturally. Serine, chemical name 2-amino-hydroxypropionic acid, a naturally occurring non-essential amino acid. It may be synthesized from glycine and is used as a dietary supplement and feed additive in biological studies and tests and in culture media. Threonine, alpha-amino-beta hydroxybutyric acid, a natural amino acid essential for optimal growth in infants and for nitrogen equilibrium in adults. Cytoplasmic equals cytoplasm, the protoplasm of the cell exclusive of that of the nucleus. It consists of a continuous aqueous solution cytosol, and the organelles and the inclusions suspended in its phaneroplasm, and the organelles and inclusions suspended in the phaneroplasm, and is the site of most of the chemical activities of the cell. Cf. nucleoplasm. Phosphorylate, the metabolic process of the intruding of phosphate group into an organic molecule. Tyro, a beginner or novice. Tyrosine, a crystallized amino acid, p-hydroxylalanine, found in most proteins and synthesized metabolically from phenylalanine. It is a precursor of thyroid hormones, catechol acid, and melanin, sometimes called oxyphenylalanine propionate acid. Adenyl, the radical adenine, a 2 term sometimes incorrectly used for adenylate. Phosphoinsintide, any number of phosphorylated inositol containing compounds that roles in cell activation and calcium mobilization in response to hormones. Transmembrane, extending across a membrane, usually referring to a protein subunit that is exposed on both sides of a cell membrane. Necrosis that results from trauma or stress also generates cellular debris that must be cleared by macrophages. In contrast to the example cited above, the clearance of this debris markedly alters the physiology of macrophages. In many cases, the debris from necrosis is loaded with endogenous danger signals such as heat shock proteins, nuclear protein, including HMGB1, high mobility group box 1 protein, histones, DNA, and other nucleotides, and components of the extracellular matrix that are cleaved by cellular protease 4. Phagocytosis of these components by macrophages leads to dramatic changes in their physiology, including alterations in the expression of surface proteins and the production of cytokines and pro-inflammatory mediators. Good day, it's Dr. Will, CBMD PhD case, Friday, May 6, 2016. It is 7:32 p.m. Eastern Standard Time. Macrophages. Macrophages display remarkable plasticity and can change their physiology in response to environmental cues. These changes can give rise to different populations of cells with distinct functions. In this review, we suggest a new grouping of macrophage populations based on 3 different homeostatic activities: host defense, wound healing, and immune regulation. We propose that similarly to primary colors, these three basic macrophage populations could blend into various other shades of activation. We characterize each population and provide examples of macrophages from specific disease states that have the characteristics of one or more of these populations. Macrophages have long been considered to be important immune effector cells. Elie Machnekov, who won the Nobel Prize 100 years ago for his description of phagocytosis, proposed that the key to immunity was to stimulate the phagocyte. Since this discovery, immunologists have been occupied with the concept of macrophages as immune effector cells and with understanding how these cells participate in host defense. However, by focusing on the immune functions of macrophages, immunologists have ignored their vital homeostatic roles, which are independent of their involvement in immune response. Macrophages are prodigious phagocytosis cells that clear approximately 2 times 10 to the power of 11 erythrocytes each day. This equates to almost 3 kilograms of iron and hemoglobin per year that is recycled for the hosts to reuse. This clearance process is a vital metabolic contribution without which the host would not survive. Macrophages are also involved in the removal of cellular debris that is generated during the tissue remodeling, and rapidly and efficiently clear cells that have undergone apoptosis. The receptors that mediate these homeostatic clearance processes include scavenger receptors, phosphatidylserine receptors, the thrombospondin receptor, integrins, and complement receptor 3. In general, these receptors that mediate phagocytosis either fail to transduce signals that induce cytokine gene transcription or actively produce inhibitory signals and or cytokines, and most of the phagocytosis that occurs on a daily basis by macrophages is independent of other immune cells. The alterations in macrophage surface protein expression in response to these stimuli could potentially be used to identify biochemical markers that are unique to these altered cells. The mononuclear phagocyte system, macrophages as recognized by Mechnikov, can be found in all tissues as circulating cells called blood monocytes, which are around 20% of the peripheral blood mononuclear cell PBMC fraction, considerably less numerous than the other major phagocyte population. The polymorphonuclear cells of neutrophilic granulocytes, Dale at 2008, have provided a historical overview of the distinct biology of these two lineages. Therefore, the primary role of macrophages is not to function as an elite immune effector cell, but instead as a common genitorial cell, the main function of which is to clear the interstitial environment of extraneous cellular material. Monocytes are recruited into tissues in response to a very wide range of stimuli with slower, rather distinct kinetics from the short-lived granulocytes. The older literature considered that they were derived from mesenchymal cells and shared biology with endothelial cells, leading to the widespread use of the term reticuloendothelial system, RES.